Good afternoon, everybody. I am Fernando Genta, researcher from Oswaldo Cruz Foundation in Rio de Janeiro, Brazil. I want to present our last work, Beta-1 glucanase inhibitors in Brazilian brown seaweed. This work was performed by Tainá Ferreira. She's a brilliant student doing PhD at our lab. And in this work, we have the help of José Barufi and Paulo Horta, which are professors in Federal University of Santa Catarina, and Daniele Castro, she's a doctor at our lab in Rio. Well, Beta-1 glucanases are enzymes which are essential for the breakdown of Beta-1 glucan polysaccharides. Beta-1 glucan are long chains of glucose residues that are bind by beta links between carbons 1 and 3. These enzymes hydrolyze these bonds, releasing oligosaccharides that are cleaved by beta glycosidases, which releases glucose. So organisms that use Beta-1 glucans as an energy source need these enzymes for obtention of glucose. Along the years, we characterized Beta-1 glucanases in several insects: cockroaches, grasshoppers, beetles, moths, termites, and lastly in vectors, in insect vectors, like the sandfly, Luxomyia palpis, and the yellow fever mosquito, Aedes aegypti. And we have shown that these enzymes are essential for the physiology of these insects. This happens because Beta-1 glucans, they are important constituents of the cell wall of fungi and plants. So insects that feed in plants and fungi need these enzymes for the breakdown of cell walls and getting access to the intracellular nutrients like proteins, nucleic acids, and storage polysaccharides. So it will be very interesting to find inhibitors for these enzymes that we can use against insects in general. So, and how people get this idea of searching for Beta-1 glucanase inhibitors. This is not a new idea. There is an important parallel in the interactions between insects and terrestrial plants. So insects that are plant feeders, they produce amylase, an important digestive enzymes that targets starch. So this is kind of, this is a cell, a plant cell. So here is the starch, a very important nutritional polysaccharide. So these insects produce amylase, aiming the digestion of starch, and in response to that, plants produce amylase inhibitors with the ultimate goal of inhibiting the digestion of the predator. A group in Russia, kind of 20 to 30 years ago, decided to investigate if a similar interaction may happen in marine environments, studying the interaction between mollusks and algae. So it happens that Beta-1 glucan is the main polysaccharide, the reserve polysaccharide of brown algae. So the idea is that these mollusks would produce Beta-1 glucanases as an important digestion enzyme, targeting the reserve polysaccharide, and in response, this brown algae may produce inhibitors, Beta-1 glucanase inhibitors, to stop the digestion of the mollusks. And the Russians were right. They found beta-3-glucanase inhibitors in several algae species. They show that these inhibitors are of proteins, and they effectively inhibit the digestive beta-3-glucanase of the predators. So we decided to investigate if the same would happen in Brazil, looking in Brazilian species. They are very different species of algae in a very different environments, and different interactions may happen. So it will be important to see if we can find the same kind of inhibitors. And in the future, if we are successful, we may explore these inhibitors to target insects, the insects that urban pests, vectors, or even agricultural pests. So we studied 4 species of brown seaweed. They were collected by Professor José Barufi and Paulo Horta in Bahia, in a city named Arraial da Ajuda. So we work with Padina gymnospora, Colpomenia sinuosa, Lobophora, and Dictyota. These 4 species were collected, dried, classified, and sent dry in silica gel and sent to Rio de Janeiro to the so we made the experiments. So we used 3 different solvents: phosphate buffer saline, absolute ethanol, and ethanol 50%. These algal tissues dried in silica, they were macerated with liquid nitrogen, and the dried powder was suspended in the 3 solvents, different batches, of course. So after suspension, these preparations were incubated for 3 days at room temperature in the case of ethanolic extracts, and at 4 Celsius degrees in the case of phosphate buffer extracts. So after incubation, these samples were centrifuged, and the supernatant was frozen and used for the inhibition assays. The assays were this is quite straightforward. We are talking about hydrolysis assays using laminarin from Laminaria digitata as a substrate. This was purchased from Sigma. This is a beta-3-glucan. And the enzyme that we use is a commercial enzyme from Trichoderma, the beta-3-glucanase. It was also purchased from Sigma, and we followed the releasing of or reducing sugars, which in this assay, in this case, we are talking about oligosaccharides or disaccharides or even glucose that were detected by the nitrosalicylic acid or bisinchonidic acid in an adapted assay that was performed in a PCR machine. So this is the kind of result that we got. So we measured absorbance a long time, and we saw activity in the controls. And when we add the algae extract, we expect to see inhibition or lower activity. And so we describe the both activities and the percent of inhibition. So the first results that I want to show are the results that we obtained with Colpomenia sinuosa. So here are different samples. We separated these samples in batches with different weights of dry tissue. These samples were extracted with the 3 different solvents: ethanol, ethanol 50%, or phosphate buffer. And in the case of Colpomenia, we only saw inhibition using ethanol 50%, which suggests that in this case, in this algae, we are dealing with an inhibitor that is probably hydrophilic in nature. So it doesn't dissolve very well in pure ethanol. Another batch of results with Dictyota, we obtained inhibition both in ethanol and in ethanol 50%. This suggests that in this case, the inhibitor may have an hydrophobic nature as well. And we didn't see inhibition in phosphate buffer. In Lobophora, we saw higher inhibitions with ethanol, a lower inhibition with ethanol 50%, and no inhibition at all with phosphate buffer. We stopped to work with phosphate buffer because we didn't see inhibition with any sample. So, but anyway, in this case, the results suggest that we are working with a hydrophobic inhibitor, which with a good inhibition activity, this inhibition is quite significant. And last, the results with Padina gymnospora, we obtained good inhibitions with ethanol and ethanol 50%, and no inhibition with phosphate buffer as expected. And these results show a very consistent inhibition pattern. All samples that we studied had inhibitory activities, suggesting that in this case, this source of inhibitor is it's more reliable. It's a more reliable source for getting beta-3-glucanase inhibitors. And again, we saw a higher inhibition activity with ethanol as a solvent, suggesting that we are dealing with hydrophobic molecules, which is quite consistent with the pattern of interaction between beta-3-glucanases and their substrates. Most of the interactions are hydrophobic in nature. So, well, discussing the results, we might consider that different solubilities that we saw depending on the algae, but in general, these inhibitors, they are hydrophobic and better extracted with ethanol. There is a controversy if we are working with proteins or sugars, but when we consider the amount of algae that we use for the extractions, it's very unlikely that we are working with sugars. We made kind of simulations, and the amount of sugars that we might get in ethanol is very low. These molecules are they have very low solubility values, especially beta-3 oligosaccharides. And if we compare our results with the Russian studies, the Russians characterized very well these extracts, showing that the inhibition it's mainly due to proteins present in these extracts. So sugars, they are not very important in these activities. And another thing that is important to consider is the heterogeneity of samples. We got from the same species very different inhibition values. This might happen because we are dealing with different parts of the plant, or it might happen because, and this was very well characterized by the Russians, we might see different inhibitor contents depending on the season of the year, on the physiological conditions of the algae, exposure to predators, content of beta-3-glucan. So these vary a lot, and this may be the cause of these differences between our samples. But anyway, the best candidate for future studies is Padina gymnospora, and this is the species that we intend to focus in the future. And it's important to mention that we try to do some inhibition assays against insect enzymes using Lutzomyia longipalpis as a model, and that was not very, we didn't get very good results because the sandflies are very tiny insects, and the amount of enzyme that we can get is very low activity at all. So in this respect, we need to work with recombinant enzymes or with insects that are bigger, so we can get higher amounts of beta-3-glucanase activity to do proper screenings. So in conclusion, is it possible to get these inhibitors from Brazilian sources? And ethanolic extracts, they are better for doing this kind of research. And we have a good candidate for further studies in the future. I would like to thank you your attention, of course, and I would like to thank you especially Professors José Barufi and Paulo Horta from Santa Catarina, Dr. Daniele Castro from our lab, and all the team of our laboratory, and our sponsors, Fiocruz, Fapergi, SINPQ, and CAPES, the Brazilian agencies that funded our research. Thank you for your attention, and bye-bye.